Okay, so just to start off, I'd like to thank ILSI for inviting me here today to tell you about some of the work that we're currently doing in Dublin. So just to start off with, I've put up here the funding agencies that fund some of the work in our laboratory and the majority of the work that I'm going to cover today. And as also mentioned, I should say that I am director of the European Nutrigenomics Organisation, which is an organisation of 28 universities and research institutes that have a fundamental interest in molecular nutrition and personalised nutrition, and I'm also secretary of the Irish section of the Nutrition Society. So what I'm going to talk about today is give you a very quick overview of what we consider nutrigenomics, and I'm going to very quickly move into talking about metabolomics, and in metabolomics in the context of food intake and food intake biomarkers, and then also in the context of looking at mechanistic studies. I will also talk about individual responses because it is a big issue in nutrition research and the huge variability we have in responses to interventions, and I'll end up then with some takeaways from my talk. So nutrigenomics, as we know, is the application of any of these omics technologies in the context of nutrition studies. And some of these are, for example, metabolomics on top, who can tell us about metabolic pathways and how they're altered under different conditions. Proteins can tell us about alteration in protein expression and protein modifications, again, under different conditions. Under different dietary regimes, for example. Epigenetics tells us about DNA modifications, DNA methylation and histone modifications. And transcriptomics can tell us about the expression of transcript levels. Application of these in the context of nutrition has really grown in the last 10 years, and it has started to help us, and we're starting to reap the benefits of those applications now. It's started to help us to understand the relationships between food and health. And I think we're on the cusp of getting really much more out of these omics technologies, and we'll start to see that, and I hope to show you that over the next few minutes of my talk. So what kind of areas can nutrigenomics help us with? Well, the first one is in study compliance. We know that this is often an issue when we're carrying out especially food-based interventions. And the application of metabolomics can help us in this. It can, if we have good dietary biomarkers or good food intake biomarkers, we can really start to look at study compliance and ensure that we have good study compliance. As I mentioned earlier, application of any of the omics techniques or multiple together can help us understand the mechanisms by which different dietary interventions are working, and it can provide an evidence base for our link between food and health. And if we employ them correctly, we can start to understand and disentangle this huge inter-individual response we have to dietary interventions, and then hopefully in the end move towards personalised nutrition or precision nutrition, as we often refer to it now. So just to go into metabolomics and nutrition and how it's being used at the moment. So I've broadly defined it into these four broad areas. There are other applications, but just these are the main ones that are in the literature. The first one is to look for biomarkers of food intake. So these are objective measures that help us with food intake, so they can help us with our dietary assessment, and I'll come back to that later. Metabolomics is also being used to do some in-depth phenotyping, and this can help us in the context of precision nutrition and understanding individual responses to diets. We can use it pre- and post-interventions and look at the alterations in different metabolic pathways that intervention caused, and this can again build the evidence base for that particular diet or that regime. And of course the last is the study of diet-related diseases. For today's talk, I'm going to focus on the first two applications and give you some examples of how metabolomics has helped us and how we can incorporate them into different study designs. So these are very broad areas, and you might be sitting there, God, can it really do all of that? It's a huge area. But if you just think a little bit about what we're doing. So we have our food intake, our food that we ingest, and our food that we ingest contains thousands of metabolites. And if we gear our metabolomics platform towards these metabolites that originate in the food, and what we call the exogenous metabolome, we can get a handle on the food intake metabolomics, or food intake biomarkers. If we then think about the food that we ingest, it can also have an impact on our endogenous metabolome. So these are our metabolites that are present in our metabolic pathways in our body. So it can impact on these. And if we home in on this metabolites, so these endogenous metabolome, we can start to understand metabolic phenotypes and we can start to see altered pathways under different conditions. But it does take a little bit of thinking about it to make sure that you get the best out of your metabolomics data. So you need to think that you're going after, if you really want food intake biomarkers, metabolites that are originating in the food. And if you want to look at how that food is impacting on metabolic processes, then you need to start looking at endogenous metabolome. So I start with food intake biomarkers then. So we heard a lot this morning about the issues that we have with measuring accurately dietary intake. And for many years, this has been the elephant in the room from a nutrition research point of view. And we know that current methods such as food diaries, food frequency questionnaires, and 24-hour recalls are all associated with a number of limitations. We heard very nicely about some of the methods that we can use to try overcome some of these limitations. But dietary biomarkers are also another way we can overcome them, or we can use them, so they can offer an objective measure of intake. Now, they won't replace self-reported data because we won't find biomarkers that cover all of the foods that we eat. But we can find biomarkers of some of the foods, or the foods that we think that we might want to track. And where the novelty, or where the growth in the area will come from, will be combining the biomarkers with self-reported intake so that we can increase the accuracy of our assessment of dietary intake. So the approach that we have been using to try identify some of these biomarkers, because it is difficult to identify biomarkers that are going to be specific, that are going to be sensitive, and are going to respond to different doses of intake. So I want to spend a little bit showing you about the approach so that you get a feel for the difficulties that is involved in it. So if we start with controlled interventions, we do these in a very controlled environment where we feed volunteers the foods that we're particularly interested in. We collect biological samples following consumption of the foods, and we identify then metabolites that are associated with the consumption of that food. We then perform our dose-response studies to ensure that the biomarker then is going to respond in a dose-responsive manner so that we can actually use it then to relate to food intake. And finally, then we move into independent studies, or studies where volunteers are free living, to try validate or confirm these in the context of a habitual diet. And of course, metabolomics underpins all of these different stages. So the first study I'm going to talk about just shows you how we go through that process of trying to identify markers that are associated with food intake. And we were interested in markers of legume intake. So we started off with test foods, and the test foods that we had in this study were carrots and peas, and we had control foods as well, and in this instance it was turnips and couscous. And the volunteers randomly ate these foods, and we collected biofluids following consumption of the foods. We then performed our metabolomic analysis. We spent a lot of time analysing the data and picking out features that were associated with the consumption of the food. So then we ended up with a panel of markers that we thought were particularly interesting related to in the food consumption, and I'm going to talk in context of pea consumption for this talk. So we can look at the excretion kinetics of these, and you can start to pick out specific metabolites that are excreted according to the test food you want by having other test foods in there and only being interested in the food that's where you have excretion kinetics or significant excretion kinetics, and the test food is going to remain pretty flat. So these are all interesting features to start with, and this is an example of one where the response is quite similar in both the test food and the control food. We then go on to identify these through databases and authentic standards to validate the confirmation of the standard of the markers. And in particular with this one, we ended up with a marker called 2-isopropyl malic acid. Once we'd found the markers, then we wanted to go on to validate them further and see if we increased the dose of the particular food, would the excretion of the marker also increase? So we performed a biomarker validation study where we fed volunteers different portions of the food in conjunction with their habitual diet. So this was done over 3 different weeks where they consumed a specified portion of the food, and at the end of the week we collected a fasting urine sample. And what you can see over here is that, yes, indeed, the 2-IPMA was significantly increasing as the pea intake increased in this study. So now we had a marker that was responding in an acute scenario. we increase the dose of it in a habitual scenario, and it also increases in the urine levels. We then wanted to confirm it in a completely independent study, so our colleagues in the University of Copenhagen had performed a similar study. They were looking at meat biomarkers, but as their control food, they had used a pea burger. So we went into their data in a very specific fashion and looked for that marker, and we could see again that this marker was associated with pea consumption. So what we concluded from this was, using our controlled environment, we were able to identify a marker, we validated it in a dose-response environment, and then we went on to validate it or confirm it in a separate study. So this is the kind of scenario that we need to do to get actual markers that are related to food intake. So it's a huge amount of effort and it's a huge amount of work to get markers that are specific to the specific food. But then the next question is, how do we actually use these biomarkers? There are a number of ways we can actually use them. And of course, the first one is, as I mentioned earlier, we can use them simply to look at adherence to specific dietary interventions. And I'll give you an example of that later on. The second one is really, can we actually use them as measures of objective measures of dietary intake? And for certain foods, there are markers where you can, and for other foods, they don't work. Markers don't work as being actual objective measures of intake. And then finally, we can look at them for relationships with health parameters. So I'm going to now just zoom in here and look at objective measures of intake and show you an example of where we actually tried to use a biomarker to actually objectively measure what people were eating. So the concept here is that you take a urine sample, you determine the biomarker concentration, and then from that biomarker concentration, you should be able to determine the food intake in terms of grams per day. So we did this using a marker that was present in the literature, that was known in the literature to be related to citrus intake. So we took fasting samples, we analysed them using NMR in this instance, and we measured this marker called proline betaine. We performed a very controlled intervention study where we fed volunteers different quantities of citrus, and we measured the excretion of the proline betaine, and we developed these calibration curves. And the concept here is that you could then take any urine sample, plug it into your calibration curve, and get an estimate of intake in terms of grams per day. So the first question then is, if you do that, well then how does it compare to what we currently do? And what we currently do is our self-reported data, so our 4-day semi-weighed food diaries. So we wanted to take our biomarker data, plug it into our calibration curves, and estimate intake, and then compare it to our 4-day semi-weighed food diaries, which give us our habitual intake, and we were hoping that we would get good agreement between these two. So just to recap on that, so we had a very detailed study where we developed calibration curves. Then we had a population study, which was the National Adult Nutrition Survey. So this is our dietary surveillance study in Ireland, where we survey 1,500 individuals, and from those we had urine samples, where we measured the biomarker, and we had also self-reported dietary data. But before I go on and show you that data, I'll just talk you through a little bit about the NAN study. So we took the participants, 600 of them, we took their food diary data, we took out the citrus intake, and we reported this as reported citrus intake in terms of mean daily intake. We took the biomarker data, we measured the proline betaine, we plugged it into our calibration curves, and we got what we called predicted intake from our biomarker data. And what we wanted to see, what was the agreement between the predicted citrus intake and what we actually got from our self-reported data. But before I show you that, I want to just go back to the actual calibration study, the dose response data. So this was done on 50 volunteers, so it was a largest number of volunteers for this type of data. So we built our calibration curves on 2/3 of the data, and on 1/3 of the data we left out, and we asked it, how well could the urine work in this 1/3 of the data to the actual intakes that people had during this study? Because these individuals in this controlled intervention studies were in metabolic suites, so we knew exactly what they were eating. And you can see here, you can quickly, this is just a snapshot of the data. This is the actual orange juice intake, and this is what we predicted from the urine samples. And you can see that the biomarker worked very well, both at low levels and at high levels of intake. And if you look at the correlation analysis, both for fasting urine sample and for a 24-hour urine sample, we have a correlation of about 0.9 between the actual intake and what we predict from the urine sample. So an extremely good correlation between both. Then if we go to our population study and see how well does it work in the free-living population, so we quantified it, plugged it into our calibration curves and estimate the intake based on the urine sample. And again, if we take a snapshot of the data, this is what's recorded here, this is what's predicted intake, and again you can quickly look down through the snapshot of the data and you can see that there's very good agreement, again both at low levels of intake and at high levels of intake. If we look at the total population using a Bland and Altman plot, we can see that the mean bias is quite low, but there are individuals where there isn't good agreement. And we would expect this with the self-reported data. We'd expect that some of the individuals are underreporting what they're eating. And in this instance, we got the biomarker data is telling us that there was much more orange juice or citrus consumed than what the self-reported data was telling us. But the overall message from this is that if you have done the correct studies and you have done a very detailed calibration studies and you know and you can quantify your biomarker in your urine sample, for certain foods you can get objective measures of intake. And if you have that, then you can incorporate these into your studies and into nutrition studies to help with things like checking and monitoring adherence. So you can really start to look at, if you know what foods individuals are eating, from the very beginning, if you incorporate it into your study, you can start looking at monitoring adherence. An example of that will be from the Predimed study, so a large randomized controlled trial carried out in Spain. And they used a biomarker called urinary hydroxytyrosol to monitor extra virgin olive oil consumption for adherence to the diet in their volunteers. And they also used one of the fatty acids to look at adherence to the walnut consumption. So these are just some examples of how you can potentially use these biomarkers to help us in our studies. So not all biomarkers will actually have a very nice relationship with intake. So some of the biomarkers will be associated with intake but won't give us that lovely dose response that I've shown you in the two examples that up to date. But that doesn't mean that those biomarkers are not useful. They are actually useful if you acknowledge that they're related to food intake, but that they are also governed by other factors. And those factors can be, for example, genetic factors, microbiome, etc. And those biomarkers then can actually be used as surrogates or intermediates to look at the relationship between diet and health, acknowledging that there are other factors that are influencing the biomarkers. And we've done some work on that, and I won't go into it in detail, but this is an area, again, that's starting to grow and has great potential when in the area of relation food with health. And application of any of these omics, of course, as I said earlier, can help us understand the mechanisms or the relationships between food and health. And the idea or the concept is that you measure them pre the intervention and you measure them post-intervention, and you look to see, can they tell you about pathways that are altered due to the intervention. And for example, you can start to get diagrams like this, which is looking at the effect of a high-fat feeding regime on liver samples from an animal study, and you can start to point out or pick out pathways that were altered due to that feeding regime. And this can then lead us on to further hypotheses that you can then follow up on. So we're starting to see lots of examples of this where the omics and nutrigenomics, if employed correctly in intervention studies, can really start to help us understand mechanisms. And for the last part of my talk, the last 5 minutes or so, I'm going to talk about individual variability. And it has been estimated that only about 40% of individuals that take part in our intervention studies actually respond to the intervention. And it's well accepted that there's high variability within the food responses or the dietary responses. And we start to now need to think about new approaches of how to capture this and how to utilize this data. So I'm going to talk about a study that we've done that started to think about this and to show you some of the potential that we have in this area. And we were interested in the role of dairy proteins in glycemic management, but we were also interested in the individual response and using omics to develop a predictive response to the response of dairy proteins. So we carried out a randomized controlled study, or randomized crossover study, examining the effects of protein drinks, and we had 3 protein drinks. We had an intact protein, a casein hydrolysate A and a casein hydrolysate B, and these 2 casein hydrolysates were just hydrolyzed in a slightly different way. The protein drink then was consumed by 20 volunteers twice a day, once with their standardized breakfast and once with their standardized meal. And we had 20 volunteers, as I said, and they were aged between 40 and 65, and they were either overweight or obese. They all wore a glucose monitor, so we monitored their glucose continuously throughout 14 days with this patch on their arm, and we read the glucose every 15 minutes. Now, the study was set up so that the drinks were drunk, each drink, so the 3 different drinks were drunk on 3 different consecutive days. So each individual, in essence, repeated the experiment multiple times. So we were interested in looking at the glycemic response to breakfast, and a standardized breakfast was eaten the whole way throughout the study. So we had the individuals repeating what they had done, so we could start to tease out the individual variability in the response due to the study design. So if we look at this in a classical scenario, we just group everybody into averages and we look at the different responses to the protein, and we do an area under the curve for the response at breakfast. We can see that the casein hydrolysate had a significantly lower area under the curve at breakfast time. However, we were very interested, and we had said this from the outset, and that's how we designed the study, to look at individual responses. And the data allowed us to look at the individual responses because each individual drink was taken 3 times for 3 breakfasts, and the exact same breakfast was taken on those 3 days. So when we started to pull out the individual levels, we started to see that individuals such as this one here on your left showed quite a different response to the 3 different proteins. Again, with the protein B showing the lowest response in terms of area under the curve. However, there were a number of individuals that it didn't matter what protein drink they were on, their response was exactly the same. So due to the fact that we had multiple readings on each individual, we were able to try out doing a type of N of 1 analysis on the individuals. And what was really interesting when we've done that was actually that only 3 individuals would have benefited from drink B, even though the average area under the curve was significantly lower for drink B. And only 3 individuals would have benefited from drink A over the other drinks. So what we need is starting to think about developing predictive models that could predict how individuals were going to respond to these different drinks using the data that we gathered. So we had also blood samples and metabolomic profiles, so we looked to see the different responses. Could we identify a profile that was predictive of the response? And we could to a certain extent, but we need to work further on this and to validate it in a different cohort. What was also interesting is that because we had individuals repeating what they had done, we were able to look at the intra and the inter-individual variability. And you can see that the inter-individual variability was very low, between 5 and 7%, whereas the inter-individual variability was up around 19%. So what you can conclude from that is that the responses were highly reproducible within an individual, but very different across the individuals. This high variability, then, we need to start trying to understand it further, and we need to start seeing how we can harness it to work towards precision nutrition. And one way of harnessing it is designing into our studies multiple measurements of the same thing, so repeated measurements, so we can look at the individual's response, both their own individual response, and then compare it to the inter-individual response. Ultimately then, where all this is going is down towards the bottom here, where we have a predictive profile based on the omics and other characteristics, so you can add in other demographic characteristics, etc. You could add in their dietary intake, etc. And based on this predictive profile, you could be able to give an individual a personalised diet. We are nowhere near that yet, and we need much more evidence-based trials up here. but we need them to take into account individual responses. So that means that rotating individuals through different dietary regimes, measuring or doing deep phenotyping of them to try and understand if we can grasp this individual variability. And of course, the omics techniques will play a big role in that, and also in trying to develop this predictive profile. So the key takeaways then from my talk would be that you can incorporate omics into your nutrition studies. They can enhance the studies, but I would say that they would need to be incorporated from the beginning. You need to think about what you need out of your metabolomics data, what type of questions you need, and then what platforms you would use to answer those questions. It can be very informative to tell us about mechanistic information, and an area that I start to see grow rapidly will be the use of these markers in terms of compliance and adherence. Of course, all of this then can lead into precision nutrition and the development of the area and trying to capture that individual response. So finally then I have to thank the team. So this is the team back in Dublin. Thank them for all their hard work. These are all the funders, our collaborators for many of the projects, and thank you for listening.